As the final lecture in this course, what I really want to do is review what we've covered in the course and then really go on and spend a fair bit of time in that review in how you might decide which is the most appropriate hypothesis test to carry out. And so I'll spend a little more time on that than other material. And in particular, there is also a table, that spans a couple of pages in the textbook on pages 366 to 368 that leads you to, really it's a table that allows you to, how you hone in on which is the kind of the most appropriate test to carry out. So as you really look at that table, it may be helpful. In part, I'm describing this because when you finish this course, what you want to be able to do is to be able to carry out an experiment, collect some data, and then use the most appropriate statistical test to analyze that data. And so this is really where we want to be when this course ends. And also, to the largest extent, the final exam will be largely hypothesis testing with only a little bit of other material in there. And then finally, I'll briefly mention some other statistical methods, in part because you might see them later on in your careers, or maybe even already. You might see more advanced methods than we've covered. And so I'll at least touch upon some of those. Some of them are really extensions of the kinds of hypothesis tests we've carried out, and some are somewhat novel things that we really haven't dealt with at all. All right, so let me first begin by saying that when we began this course, where we really started was looking at data. And we did this in a couple of ways. And really, despite the fact I really haven't emphasized this a lot, really whenever you collect data from an experiment, the first thing you do is you really should look at it. And the way to look at it is to, graph it in some appropriate way. So we spent a fair bit of time on how one should graph data. And in part, that depends on the kind of data. Is it categorical data? Is it numerical data, etc.? How many variables are you characters or variables are you measuring? That might influence how you're graphing it. And there's quite a good chapter in the text that, you should refer back to when you leave this course that really gives you really good ideas on how to present different kinds of data. So I'd hang on to that, at least that chapter of the book, as being very useful. I often look at it if I'm going to publish stuff as well, because it's really quite helpful. So we can graph data in some appropriate way with histograms or bar plots or etc., scatter diagrams. And also we can produce descriptive statistics. In other words, those summary statistics like the mean, the variance, the standard deviation, coefficient of variation, etc. Right? All those descriptive statistics are also very useful to look at as well. And you can run SAS to get out all of those using proc univariate, for example. So that's all I'm going to say really about this, so I'm really spend very little time on something that is perhaps one of the most important aspects of the whole analysis process, and that is really looking at your data. When you look at your data, you might get some new biological insights. You might see some unusual features. You might quickly notice there is going to be some violations of assumptions of hypothesis tests you're going to do, and you need to ameliorate those somehow with transformation or using non-parametric tests. So many reasons to have a really good look at your data before you do anything else at all with it. Okay, so that's it for that portion. And so now what I want to talk about is really how you go about deciding which hypothesis test to use. And as an overriding feature of this, or the driving feature of this, really is you always want to use the best hypothesis test. Obviously, it has to be one that's testing the hypothesis you're spelling out, that's embedded in the biology you're doing. And remember, there's a difference between a biological hypothesis or a biological idea or a biological question and a statistical one, right? We use the word hypothesis in two different ways. You may well have some biological hypothesis or question or idea that is framed in terms of biology, right? The word null hypothesis shouldn't and won't appear in there at all necessarily. It's only after you translate that into statistical gibberish that you then have a null and alternative hypothesis. So which hypothesis test are we going to use? And here I'm really referring to the statistical hypotheses. So there are a number of ways of factors that you need to consider when you're making this decision. So one of them is, what's the biological question you're asking? So you'll have some biological question, that you're going to ask. Is one thing bigger than the next? Are you comparing means, for example? And if the biological question involves, is this bigger than that? Is this thing faster than that one? You're going to be comparing the means of the speed of fish growing one way or another, etc. So that's going to already be one of the clues. Am I going to be comparing means? Or you might be asking questions about variability. Do I want to know if something is more variable? Am I comparing variances? Right, so this is what. Am I asking whether two or more things are associated? And then, there'll be other sort of issues, and that is how many groups am I comparing? Or how many treatments am I applying? Do I have a control and then some other number of other, concentrations I'm growing things in, or applications of a herbicide, or different pHs, etc. Right? How many treatments am I applying? So these sort of issues will be or are going to be embedded in the biology you're carrying out. What is it I'm asking about? I'm asking about variances, the size of things, in which case it would be means. Are two different characteristics associated? So that's one thing you need to think about, take into consideration when you're going to carry out, when you're trying to decide on what the most appropriate hypothesis test is. The next thing you need to, or another thing you need to consider, doesn't have to be done in this order, but the nature of the data you're going to collect. So by this I mean, are the variables categorical? Are they numerical? How many variables are there? Because you could have, say, two variables, one might be categorical, the other numerical. You might have two variables, both are categorical or both are numerical. So those are important issues in deciding ultimately what you're going to do. Are the data actual measured quantities, like pHs or the weights of things or heights of things or rates of oxygen release? And are they measured values or are they ranks? And if the data are ranks, right, remember ranks are simply you line individuals up from smallest to largest and assign a rank, 0, 1, 2, 3, 1, 2, 3, 4, 5, 6, and so on. Because all these things are going to lead you, really will help you exclude certain kinds of hypothesis tests. So for example, if my data are categorical, if I have one variable and it's categorical, let's say I have one variable that's categorical, I know I'm not going to be doing a t-test. I'm not going to throw out a lot of statistical hypothesis tests because those many tests apply only to numeric variables. If my data are ranks, then we've only covered one, really, though there are other ones we haven't covered. We've only covered one test that involves the analysis of ranks, and that's the Mann-Whitney U-test. Right, so you would go directly to that. I mean, you obviously want to read what is the question, and just to make sure everything lines up and it is the appropriate test. Okay, and so what else do we need? We need to know, we might want to also take into consideration the assumptions of particular tests, because we want to know, have we met those assumptions? Right, so just to go back again, really what's going to help you get to which is the hypothesis test is what's the biological question? Am I asking about sizes? Am I asking about variability? Am I asking about the association between characters? How many treatments or groups do I have, right? If you have just two groups and you happen to have numerical data, you might be in the realm of it's about means, and you're going to be maybe independent sample t-test. And if you have more than two groups, it's not going to be a t-test because t-test can only be done on two groups. You want, again, the nature of the data, right? We've probably learned roughly an equal number of tests for categorical data versus numerical. So if your data are all categorical, you can throw out half the hypothesis tests we learned, and now you can hone it down to those. All right, so again, these are really all the kinds of questions you should be asking yourself, or that will help direct you to what is the most appropriate test. So that's an approach you should take, and you, again, look at the table in the book that will help direct you. So let's just now go over some of the kinds of tests and kinds of data we had. So we had categorical data. Remember, this is like count data where you classify individuals. Some number of individuals have red eyes, some have purple eyes, and some other number have green eyes. And those are categorical data, right? You're categorizing every individual in your data set by eye color, let's say. So in the categorical realm, we had goodness of fit tests. So these commonly crop up in genetics, for example. Right, and so we were going to do a chi-squared test against expected proportions. Where recall that often, certainly in genetics context, the expected proportions come from Mendelian ratios, for example. We also did chi-square tests against expected distributions. And so the one we did a lot of was the Poisson distribution. If something is randomly distributed in time or space, it should follow the expected Poisson distribution. We did binomial tests. We can, instead of a chi-square goodness of fit test, we can always do a binomial if we have just two categories, only two categories. And we just arbitrarily decided that if the total sample size is less than or equal to 25, and you have two categories, do a binomial test, right? And the reason I've suggested keeping it to 25 or less is because you have lots of calculations to do. The bigger the sample size, the more heavy number crunching involved by hand to do a binomial test. Ideally, whenever you have two categories, it would always be best to do a binomial test because it gives you the exact p-value, as opposed to the chi-square test, which is an approximation. So we're still in the realm of categorical data. If you have two categorical variables, so just for example, if you random sample a bunch of individuals and you record their hair color, green, purple, or orange, and whether they're male or female, then you have two categorical variables. And you can ask whether they're associated, so that's a test of independence. Okay, so if you have nothing but categorical variables, we're in this sort of realm of goodness of fit tests, where you might be testing against some expected proportions driven by genetics predictions, or by sex ratios might be equal, or by expected distributions of things, such as the Poisson distribution is something randomly distributed. Or you can use the binomial test if you have just two categories and a small sample size. If you have two categorical variables and you want to ask if two things are independent or if they're associated, you're going to do a test of independence. And this we did last day. So let's now think about what if you have just numerical data. We can ask questions about means. Right, and we had a one sample t-test, for example. If you have only a single sample, and the question is, does the true mean equal some predicted value? The number of grams of butter in a package or something like that. supposed to be 20, you could take a random bunch of butter pads and measure them, measure their weight, and test this hypothesis. It can be one or two-sided. If you have two independent samples and you want to compare means, you're in a two-sample or independent sample t-test. You could be one or two-sided. Remember that if you're doing a two-sample t-test, you want to first test the assumption using that the variances are homogeneous. So start with that f-test first to test this assumption. If that assumption passes, in other words, if you don't reject this null hypothesis, then do the t-test. It's the question of interest. You might have a paired t-test. So if the experimental design is such that individuals are matched in pairs by some criterion before the experiment is conducted, and one member of each pair is given one treatment, the other treatment, or one is the control and receives the treatment, then it's a paired t-test. In the paired t-test, you're really going to analyze like it's a one sample. You're going to analyze it as if it's a one sample t-test by analyzing the differences between the performance of pairs. And so in the realm of these t-tests, where we're asking questions about one mean or two means, in the case of independent sample t-test or the paired t-test, remember for these two things we're going to check out the assumptions. So whenever you're doing an independent sample t-test, test for homogeneity variances first using an f-test. For the paired t-test, the assumption is that the distribution of differences is normal. Since you can't really test that sitting there at your desk and while you're writing your test paper, instead what I want you to do is inspect the differences and look for outliers. If you find outliers, use the sign test. So you can use the sign test if differences have outlying values. Remember, the sign test really, instead of analyzing the differences, you just write down whether the difference was positive or negative, and then you're going to do a binomial test, counting the number of positives. The number of positive signs should equal the number of negatives, which should be 0.5. So you're basically going to do a binomial test with P equals Q equals 0.5. For the independent sample t-test, if the f-test assumption fails, in other words, if you reject the null hypothesis and the variances are different, then what you want to do is the Mann-Whitney U-test. So what you do is you take all the data from the two groups, rank them from small to large, assign a rank, check for tied ranks, then you analyze those ranks using a Mann-Whitney U. All right, so there's, in this portion of the course, this is really more approaches the kind of issues that you really need to face when you're doing statistics, right? You're going to look at your data first. You're going to decide what the most appropriate test is. You're going to test that you've met the assumptions of that test, and then make a decision. Have I met the assumptions? If so, you happily do that test, like a two-sample t. If the assumption fails, then you do an alternative test, a non-parametric test that has fewer assumptions embedded in it. So that was really asking questions about means. But if I have two samples, I can also ask a question about variances. Is one thing more variable than the next? So that's that f-test that I just talked about in the context of testing the assumptions of ANOVA. But sometimes the biological question is a question about variances, and so you're just going to do an F-test. Right? You can do a one-sided or two-sided F-test. Is this thing more variable than that? If that's the question, you're going to do an F-test. What if I have two or more new samples I want to compare? Means. So here I can use a single factor or one-way ANOVA we just relatively recently talked about. I'm going to use analysis of variance to compare those means. If it's fixed effect ANOVA, I can follow it with Tukey's Tukey-Kramer test to ask which means differ. If it's a random effect ANOVA, I don't really care which ones differ, but I could estimate the variance component due to whatever the treatments applied were. So that's what I might do. So again, questions about means, but this time I've got more than two means. Typically, if you have two means, ANOVA will work, but it's probably easy to do a t-set test, so I do that. If you have three means you're comparing, you want to do an ANOVA, and you may, if you reject a null hypothesis, you can follow it up with whichever of these are appropriate. And I guess I should just, as a reminder, for both t-tests and for ANOVAs, we really have two variables. One of the variables is the response variable, and the response variable is going to be numeric. Right, if I'm measuring the growth rate of saplings, it's a numeric variable, it's the response variable. I'm interested in whether different nutrient concentrations affect the rate of growth of these saplings. And then I really have a categorical variable as well. And that categorical variable typically is going to be what are the treatments. So one of the values of the categorical variable can be whether it's the control group, whether it was nitrogen applied, whether it got phosphorus, whether it got potassium. So I have a categorical variable that describes let's say the fertilizer applied. The same is true for t-test, right? I have two groups. I'm categorizing individuals as to whether they fall into group 1 or group 2, but then that's the explanatory variable, right? So this is I'm trying to explain growth by fertilizer. The other one is the response variable. So it's important to know, which one is response, kind of is it a numeric or not, is it categorical? All right, I can also ask questions where I have two numerical variables. And so the kinds of questions we've learned with these really are two things associated with each other. I've got two numeric variables, so I measure two characteristics on each individual in my sample. So if the simple question is, are two variables associated, and they're both numeric, then you're going to do a correlation. And remember, for correlation, there is no dichotomy between the response variable and the explanatory. You can just happily flip-flop the x and y axes. It makes no difference to you. Are these two things associated? And most of the time, I think I've said this as well, we use correlation too often, or many people do. You should probably typically be using linear regression, where one is a response variable, the other is the explanatory. Use linear regression. Okay, so for linear regression, we're really trying to predict one thing by the other. So I've got two, again, numeric variables. I've got a response variable that's numeric. And I've got the explanatory, also numeric. right? The explanatory is going to go on the x-axis. It's always going to go there. And the other is going to go on the y-axis. And so I might want to predict, I think examples like blood pressure by age, I think is one I gave you. Or blood pressure by, it could be some other, caffeine intake or something like that. Grams per day or milligrams per day or whatever. And we haven't learned how to handle violations of the assumptions, although I've told you how to look for violations of assumptions. So, if you plot some points and they're curved, linear regression, which is the one we learned, is really not appropriate, but you could try to straighten this out with transforming the values of the x or y or both in some way. So you can do that on computer and hope to achieve linearity. You may or may not. Difficult to do by hand because you never know which transformation will necessarily work. All right, so that's sort of an overview of this things we've covered with respect to hypothesis testing and the things you need to think about in order to get you at what is the most appropriate test to do. Right? And so you might even want to, in your cheat sheets that you can bring again, one two-sided piece of paper, you can write anything on it. You might want to have a little table or some for yourself that you draw up as to how you're going to get to which is the appropriate hypothesis test to carry out. Again, kind of data. Is it just one variable? Is it two variables? Am I asking about associations? Am I asking about means? Am I asking about variability? Right? All those pieces will help you exclude some of the tests and lead you to the most appropriate one. And finally, I just wanted to briefly mention some other statistical methods that we haven't covered just in passing, and then there are numerous statistical methods. And some of these, as I said, are extensions to the kind of things we've already done. So we did linear regression, often called simple linear regression, where we're fitting a straight line. But you could also fit curves. So remember, we had something of the form yi equals an intercept plus a slope. And there's an error term. I'll put it over here for the moment. If we wanted to fit a curve, like a quadratic form, like a parabola, we could add one more term here. So I'm just adding to my linear model an intercept, a linear term and its regression coefficient, and then a squared term, a quadratic term. And we can fit curves that way, or we can fit a fancier curve by putting a cubic term, etc. So there's this. When you begin fitting curves, and there are other approaches to fitting curves, when you begin fitting curves, you're often asking questions because you don't know beforehand what the appropriate model is. Should the cubic term be included or not? So there become some other issues in terms of testing hypotheses about whether does this term contribute anything? And normally what we do is we choose the simplest model to fit through some data. So there are some other issues involved in drawing up models that are more complex than a simple, the model for simple linear regression. So this is, as I said, polynomial regression. It's the single x value you're measuring. So this might be, the concentrate pH, let's say. If I had the linear form of pH, the square of pH, the cube of pH to predict growth rates or something. But there is also something called multiple regression. So in multiple regression, we have one response variable, just as we have in simple linear regression. So there's just one response variable. It might be growth rate of frogs, let's say. And instead of trying to predict it by one explanatory variable, you might have two or more. So I might want to, explain the growth rate of tadpoles as a function of oxygen consumption, or O2, let's say O2 concentration in the ponds they're in, UV penetration of the pond, temperature of the pond or the tank they're in. All right, so here I've got three explanatory variables. And so we can do linear regression where I've got y, that is the growth rate of the frogs or tadpoles, equals some intercept plus some term and its coefficient for O2, plus another term for UV and its associated regression coefficient, plus another term for temperature. And you can even have interactions among these different factors. So there are more complex models, and there are more assumptions embedded in these kinds of analyses. They're somewhat trickier to carry out. So this would be like I can't draw it, but like if you had just two explanatory variables. Here's the y-axis, and you might have O2 concentration here and UV irradiation amount here, and you might see some surface in space that describes the trajectory of frog growth as a function of those variables. So multiple regression. There are some analyses referred to as called analysis of covariance, or ANCOVA. And so ANCOVA can be viewed in a couple of ways, but one way is it's like an analysis of variance, but you throw in with it a variable where you control for some other feature. So it has sort of the qualities of both regression and analysis of variance. So for example, if I wanted to compare growth rates, so growth rate is my response variable of some organism or another, as a function of light. So I categorize individuals as to, they get light level A, B, and C, and they're grown at different temperatures, temperature 1, temperature 2, temperature 3. So this is a kind of ANOVA we haven't considered. It's a two-way ANOVA. But in addition to that, I might measure each individual before the start of the experiment. So measure their height at the beginning, and use that, because you think that might affect the outcome of the experiment, you use that as a covariate. So that's where the word analysis of covariance comes from. I'm trying to control statistically for the initial height or weight or something of individuals. So you might come across those. Then there are other methods called multivariate methods. So these depart from the kinds of things we've been doing. Multivariate, so far in all the things we've considered, we've really typically had just one response variable. So when I'm doing an analysis of variance, I'm measuring the height of individuals, the weight of individuals, the blood pressure, whatever it is. It's one response variable. In multivariate analyses, you have many, or more than one, many response variables that you simultaneously measure. So we haven't considered that at all. But you might come across that. It's particularly common in some fields. And so, for example, let's imagine and you might often see it in various fields of biology. Let's imagine you wanted to compare the males and females of some bird species. Well, males and females might differ in many ways, right? And so what you might simultaneously do is measure the weight of the birds. You might measure so you're going to measure the weight of a bunch of males and a bunch of females, beak length, wing length, color. You might somehow try to figure out a way to measure color as a numeric variable. So you have a bunch of numeric variables that you measure on each individual in your data set, right? Everybody is measured for all these four variables. You have a number of males and a number of females. Then you can simultaneously ask whether or not individuals differ for this host of characteristics. So multivariate methods. And there are things, I don't know if it's worth throwing out the name of them, except to say, often when you have numerous response variables, it's very difficult to look at that data and to analyze and understand it. So often people will then use a method for reducing the data down to a simpler form in something that's called principal components analysis. Or factor analysis, where you can essentially, if you have measure many characteristics, many response variables on individuals, there's mathematical statistical methods for reducing the number by reconstructing new synthetic variables that you extract statistically from the data that help you explain it. So I'm not going to go on and describe this. It's beyond the scope of what we're doing, but not beyond the your ability to understand it and down the road at some point. So there are many other methods. Let me just talk about some other, sometimes referred to as computationally intensive ways of testing hypotheses. And one that you might have come across is called bootstrapping. So what we've done really throughout this course is we've doing a hypothesis test. Let's say we're doing a t-test. We construct a test statistic, a t, and I call them t subscript calc, a test statistic, and we want to know, is that test statistic unusually large? And so we should reject the null hypothesis. And to find out whether it was unusually large, what we did was we looked it up. We looked that value up in the theoretical t distribution. So we had an assumption that the statistic we were interested in could be followed the theoretical t distribution. Well, there are methods that one can use, and they're being increasingly used, where you don't have to ask the question whether you follow some theoretical distribution, and those are often called resampling techniques or randomization techniques. So there's no assumption about this thing having to meet the t distribution or the normal distribution or the f distribution. And so these are one of the ones that you might encounter. It's called bootstrapping. And with bootstrapping, you resample, you have a bunch of data from some experiment. You resample the data randomly with replacement. And effectively what you then do is for each of these random resamplings of the data, you might compute the test statistic. It could be a t-statistic, but then you do that numerous times on a computer. So you might do this, let's say, 10,000 times. I take my data, and you need a sizable data set. I randomly sample it, I compute a t-statistic. I do that again and again and again and again and again. I can then plot the distribution, and I can effectively plot the distribution of this t-statistic I've calculated. So this would be a histogram. I've drawn in a smooth curve, but it would be a histogram generated from my computing random resamplings of my data. Then I can take the real data, the data I have, calculate the test statistic, the t-calc, and see where it falls in this distribution. If it fell over here, I can calculate this probability here, or the proportion of random samples that are larger than that, as big or larger. And that proportion is effectively, or approximates, a p-value. So I can get a p-value out without making any assumptions about what the theoretical distribution is. I'm generating, or I'm approximating, the theoretical, the sampling distribution, the test statistic distribution, by resampling my data at random. So this is done increasingly. It's especially done for statistics that are, so let's say, were recently developed, or where the underlying theory for what the distribution should be is not known. So you may come across these, and in particular you may see these in, let's say, modern or more recent population genomics or population genetics in that field. You certainly see this in the field of phylogenetics. So phylogenetics is the reconstruction of evolutionary history, where people will draw, sorry, let me get rid of that, where people reconstruct evolutionary history. So they're drawing phylogenies or cladograms, right? And this is the phylogeny of some group of species. Here's species 1, species 2, species 3, species 4, species 5, and so on. Right? You collect a bunch of data. Increasingly now, the data are DNA sequences. You sequence some number of genes. You use all that information in order to find out which species are most closely related to each other. And so one question is, how reliable is this particular branching pattern, right? This suggests that species 1 and species 2 share a more recent common ancestor with each other. In other words, they diverge most recently from each other than they do with species 3. And so what you can do by bootstrapping is effectively apply confidence intervals, essentially, to how reliable is this branching pattern, or do we really not have enough evidence to say that these two things split more recently than 3? In other words, is this branch wrong and really this one effectively sits here? We don't know. We don't have enough information to know whether or not which one of these split off first. So I haven't put a lucid explanation of this, but you might often see bootstrapping applied to phylogenetic data. So that's just one kind of random resampling technique that you might see, or computer-intensive resampling. There are other issues that I think mentioned probably in day 1, that there are really two aspects, sometimes viewed as two branches of statistics. One is hypothesis testing, the other is estimation. We've spent most of the time hypothesis testing, but of course you have to estimate things as well. And we've talked about one form of estimation. How do you get the best estimates of some value? And we talked about least squares estimation when we described how to get the best fit line through points. Now, I didn't do I didn't do the calculus to show you how you derive the best fit line or the equations for it, but it involves minimizing squared differences between some line and the actual data points in the vertical direction. Another kind of estimation that you'll often see or hear about, and that in fact we often do, even if we don't use the word, is called maximum likelihood estimation. And again, you'll see this in the context of certainly of genetics, population genetics, and phylogenetics as well, maximum likelihood estimation. And it's a method for deriving the best possible estimates of some parameter given the data. And so, I won't, sorry, let me get back to that, I didn't mean to drop out of there. I won't give a complete description of how you do it, but basically what we're doing is you collect some data from an experiment, you want to estimate something. And so here we have on this axis the parameter we want to estimate. Let's pretend for the moment this is the degree of inbreeding in some species, how much inbreeding is going on in some population, and we're trying to estimate it. And on this thing we have a quantity that we can calculate called likelihood. And so here the amount of inbreeding might vary between 0 and 1, where 1 would mean completely inbred. Every individual, if it was plants, is completely self-fertilizing, or has been for a long time. And here there'd be no outbreeding and no inbreeding. 0 inbreeding means everyone has been randomly mating for some time. Okay, so likelihood is a quantity we can estimate from some data. So we might have some genetic data on frequencies of genotypes in a population. Right, if everybody is inbred, there's going to be no heterozygotes, for example. If everyone is outcrossing, there'll be some, but we can predict how many it is. In any case, I won't go and elaborate on this except to say there's a quantity we can calculate called likelihood. And if we plot the likelihoods against all possible values of inbreeding, what we want to find is the maximum likelihood. What's the maximum likelihood? Whatever that value is, what's the highest point on this likelihood curve, that is taken as our best estimate of the amount of inbreeding. So that's the value we decide is, okay, that's our best estimate of the amount of inbreeding in this population. And in fact, the curvature around that point is an estimate of the standard error, how variable that estimation is, the standard error of that estimate. So that, or it's negatively related to it, or reciprocally related to it. So we can get out an estimate and its standard error using maximum likelihood estimation. And as you can probably guess, for those of you I think you've all taken calculus, the way to get these is first you need to specify the likelihood equation, and then you're basically doing calculus, right? You're finding the peak of some curve. So there's a whole bunch of methods. Many of the methods we use to obtain estimates are already maximum likelihood estimates that somebody has figured out. If you're doing some fancier things in genetics, let's say that you've developed in population genetics, you want to estimate things, you might have to do some more fundamental work to specify the likelihood equation and work these things out. In any case, I'm just touching upon these. I don't expect you to know or reiterate any of this material on these newer methods. I'm simply mentioning them in passing because you might encounter them at some point in your careers. Right? So you're not responsible for this material on the test. It's just some kinds of issues that might arise and some methods you might in fact do at some point if you haven't done them already, either knowingly or unknowingly. All right, so I'll stop there.